So we can, expose them to sound, and if the promoter works, then we should get GFP produced. So we look at the. They do. They glow green under UV light. So we have a cool little flashlight that we use. That is actually supposed to be for looking for scorpions, but it works for our purposes as well. So then right now we're just doing very broad testing. We're not really doing quantitative analysis. We're just seeing like yes, no, are they glowing or not. But we will eventually get into a more quantitative. Okay, good. I had it right.
So how, you mentioned briefly that this project's supposed to ameliorate the process of getting stuff out of cells. Is that how this project relates to synthetic biology?
Well, I think the project relates to synthetic biology in a number of ways. I think that anytime you are manipulating genes, you're doing synthetic biology. So the fact that we are taking genes and rearranging their order, even if we're not actually changing the sequence, I think it's still synthetic biology.
So I wanted to ask you another question about that, comparing this situation to doing just research under a professor or a grad student. Have you had experience doing that yet? Or?
I worked briefly in a biomechanics lab that was studying the origins of flight. So they were making models of dinosaurs, the first flying dinosaurs, or the first winged dinosaurs, and testing them aerodynamically. And my part in that project was very small. All that I was doing was gluing feathers onto the models and sewing the wings together. And it didn't feel like research. It felt more like knitting. And it was definitely a part of somebody else's project and not my own. And I think when you have ownership of the project, that you have a lot more pride in what you're doing. And that's, yeah.
So you think that's what this experience feels like? You feel like your ownership is?
I do. I feel like it's our project, and we're the ones who are expected to do most of the troubleshooting and to figure out what works and what doesn't. And certainly we get a lot of help from grad students and from our advisors, but I think at the heart of it is what we're doing, what we're putting together.
Do you think you would want to do, what do you think research would be like in the future if you want to continue with stuff like synthetic biology?
Well, if I were to continue with synthetic biology, let's see. Let me think about this one for a minute. All right. So I think if I were to continue with synthetic biology, that a lot of the techniques that I've learned here would be applicable. And, we really do, the full gamut, I think, from extracting the genes and doing the sequencing and learning, how to analyze the data that we're actually getting, how to look for the information that we need in research articles. And I think all of that would relate to future research, where I'd either, go to grad school or go into industry and work in a synthetic biology field. Okay. I haven't done a lot of interviews, so let's see how good I am at thinking on my feet.
I've been on this side, so I'm sorry.
Oh, it's okay.
All right, so do you think there are any ethical questions related to your IGEM project?
The biggest ethical concern that I could see with what we're doing is that we do use antibiotic resistance genes in our screening process to screen for the correct colonies and to make sure that we're actually making what we think we're making. So we have to be really careful that nothing gets thrown out, that we don't throw out living cells that carry these antibiotic resistance genes. So we have to make sure that we're cleaning everything properly at the end of the day. But as far as like the actual genes that we're making, I don't see a big ethical concern there. I don't think that what we're making is something that if it were to, I don't think we're making anything that could be used to harm someone or to do any damage like that. I think that the biggest concern is the antibiotic resistance, and that's the one that I worry about the most.
Do you know where this is a question maybe you had, do you know where those, when you clean out things appropriately and get rid of that stuff, where does it go?
Well, when we're done with our cell cultures, they just get bleached and washed down the drain, and that's it.
Oh, so they get killed?
Yes, we kill them. So we pour like a bleach solution over our plates and our tubes, and then they sit for 15 minutes to make sure all the cells are dead, and then we just rinse them out. There's a lot of bacteria that goes down the drain every day, and as long as it's dead, we should be fine. Things that we can't bleach are autoclaved, so they're heated up to a temperature that's high enough to kill anything that's alive, and then they're thrown away.
So, what was your trajectory to synthetic biology? How did you become interested in it? Like, maybe like synthetic biology starting from bioengineering. How did you?
Well, the part of synthetic biology that most attracted me was using synthetic biology to, let's see, I'm looking for the right word here. I think what attracted me to synthetic biology the most was doing things like gene therapy. So people who have certain mutations in their genes that cause problems like Parkinson's disease could potentially be cured or at least helped by using, by taking the genes and fixing the mutations. And, I think that that's still in its very early stages. I don't think that there are actually any gene therapy. I could be wrong, but I don't think that there are any gene therapy products that are commercially available yet. But I definitely think that within the next 10 years, we'll be seeing a lot more of that.
Why did the IGEM team move upstairs?
Well, in the lab that we were working in before, we were having quite a bit of contamination problems, and we went through a lot of different, testings of our enzymes and of our media and of our plates to see if we could determine what the root of our contamination issues were. And we had a lot of potential things that could have been causing the problems, but we only have a couple of months to finish this project, so rather than continuing trying to troubleshoot everything, we moved to a different lab. We moved upstairs to Chris's lab.
So how would you compare this research to working in marketing?
So I am a re-entry student. I'm coming back to school after working in a marketing firm, mostly because I don't think that marketing contributes anything useful to the world, whereas I think that synthetic biology, has great potential for medical uses, for producing things like insulin, that can help diabetics. I think that there's a lot of chances to change the world and make it a little better in synthetic biology, which is something that I never felt like I was getting out of marketing.